Hello students, in previous part we have studied about introduction of cell, and in this part we will discuss on prokaryotic cell, prokaryotic or prokaryotic cell. As we have discussed, the prokaryotic cell in which there is no distinct nucleus membrane present, no nucleus membrane is present, that cell is called prokaryotic cell, such like as bacteria, mycoplasma, PPLO, pleuropneumonia-like organisms. These cells, cyanobacteria, the blue-green algae, they don't contain any nucleus membrane, so they categorize into prokaryotic cell. Although the different organisms have different shape and size of cell, but the ultrastructure of the prokaryotic cell is similar in all of organisms. Firstly, we draw the structure of prokaryotic cell. This is an oval shape prokaryotic cell. Although we know that there are different shape and size of the cell, first we will take the oval shape prokaryotic cell. The outer membrane is called cell membrane. Just inside the cell membrane, there is no nucleus membrane present. So, a fluid which is filled here, that is called cytoplasm. This is cytoplasm. In prokaryotic cell, the outer membrane, which is known as cell membrane. Actually, the outer membrane is made up of three layers of different cells. First one is cell membrane, and cell membrane is covered with another peptidoglycan layer, that is called cell wall. Prokaryotic organism contain cell wall because this, due to presence of this cell wall, they have rigid structure. So we can say that cell wall provide a rigid structure to the prokaryotic cell. This is the cell wall, which is made up of peptidoglycan. This cell wall provide rigid structure to the prokaryotic cell. Cell wall. Cell wall is made up of peptidoglycan. Just outside the peptidoglycan or cell wall, some gelatinous cells are also present here, which makes another outer membrane. This gelatinous cell membrane contained, or we can say that this membrane help in adhering the prokaryotic organism with the solid spore. This is called a capsule as well as a glycocalyx. Glycocalyx. In this glycocalyx, some gelatinous cells are also present. This is the ultrastructure of a prokaryotic cell. Cell membrane inner side in the cytoplasm. At some places it forms an irregular folding. They make some irregular folding. These irregular foldings help the cell in various functions, and they are called mesosome. the cell membrane inner side into the cytoplasm produce many folding structure. These folding structures are called mesosomes. There are many important role of mesosome. Firstly, the mesosome help in respiratory. They help in respiration of prokaryotic cell. As they take part in respiratory process, they produce energy and they help this energy store in mitochondria. It means the mesosome act as mitochondria. As we know that mesosomes in the cytoplasm inner membrane means cell membrane forms many folding structure, and these folding structures are called mesosomes. Mesosomes help in respiration. Due to help in respiration, they produce energy, and so they act as a mitochondria. Number third, the mesosome help in cell division. So there are many role of mesosomes. In prokaryotic cell, DNA is present in circular. DNA is present in circular. This circular DNA, actually there is no nucleus membrane. So all the nucleus substance are scattered with cell organelles all over the cytoplasm. There is no specific space for the genetic material. That the genetic material DNA may be present in cytoplasm in circular loop. But at the time of cell division, this DNA comes closer to the mesosomes, and we can say that attached with the mesosomes, because the mesosomes help in cell division. As the cell division can take place, this DNA can pass into the another cell. So the genetic material may be transferred. During the cell division, DNA comes closer to the mesosomes. There are some ribosomes are also present in cytoplasms. As we have discussed, there is no specific place for each organelles. All the organelles are scattered all over the cytoplasm. These are ribosomes. In some prokaryotic cell, there is an extra circular DNA present rather than this one. There is some extra circular DNA is present. Suppose this is the extra circular DNA, and this extra circular DNA is called plasmid. Extra circular DNA. If some prokaryotic cell contain extra circular DNA, that is called plasmid. On the basis of plasmid, we categorize the prokaryotic cell in two types. Donor cell, it means the male cell. Or if the plasmid is not present, then that is called recipient cell, means females. Plasmid present. If the plasmid is present, then that is called positive strained. Positive strained. It means this is the donor cell. Or we can say that male cell. It is the male. If there is no plasmid present, so we can say that plasmid absent. Plasmid absent. Then that is called negative strained. And this is female. Recipient cell, and that is the donor cell. Male is donor and second one is recipient cells. And the plasmid, on the basis of plasmid present or not, so we can categorize fully, distinctly, this is male or female. If plasmid is present, there are some large finger-like projection may present. Large finger-like projections. Large finger-like projections. These large finger-like projections may present only those prokaryotic cells which contain plasmid. These are called sex pili. The sex pili only present in those prokaryotic cells which contain plasmid because they help in the conjugation and transfer the genetic material from one cell to another cell. So they are called sex pili. There are many small finger-like projections also present in prokaryotic cell all over the body. These are called Fimbris. If Fimbris are present all over whether the cell is contain plasmid or not, but small finger-like structures are present in both prokaryotic cells. These small finger-like projections are called Fimbri. Fimbri. What is the role of Fimbri? This provide adhesion. It means they help into attach with the solid spot substratum so that the prokaryotic cell may be stick at the solid spot and they cannot be able to move. Fimbri is the small finger-like projection. These fimbri are present whether the cell is the plasmid contained or not. But sex pili present only on those prokaryotic cells which contain plasmid because they act as copulatory organs. They help in conjugation the genetic material from one cell to another. Some prokaryotic cells are motile and some may not. There are some prokaryotic cells which move from one place to another place. If those cells which move from one place to another place, one large hair-like structure also present. And that large hair-like structure is called flagella. This is called flagella. If the prokaryotic cell is moved, it means move from one place to another place. They move with the help of flagella. Flagella is large hair-like structure. This flagella contains three parts. The long ribbon-like structure is called filament, and another, the attachment structure with the cell membrane, this is called basal body, and the rings which are present between the basal body and filament, this is called hook. So we can say that a flagella only found in those prokaryotic cells which move from one place to another place. A flagella consists three parts: filament, a long hair or ribbon-like structures. The basal attachment with the cell membrane is called basal body, and the connection between the basal body to filament, this is called hook. In this hook, there are many rings with the help of flagella attached with the prokaryotic cell. In prokaryotic cell, ribosomes are present 70S as sedimentation coefficient. Sedimentation coefficient. As we know that ribosomes, whether the cell is prokaryotic or eukaryotic, they are not membrane bound organelles. In prokaryotic cell, ribosomes contain two subunits. There are two subunits. One is large and another one is small. In prokaryotic cell, one cell which contain 50S ribosome and small subunit contain 30S ribosome. Suppose This is the large unit and this one is the small subunit. Both the subunits are separate from each other, but in presence of magnesium ions, these subunits are attached, such like this one. One is large, another one is short, small subunit. So this is the 50S ribosome, this is 30S ribosome, but they make 70S ribosomes. 10S sedimentation coefficient may be eliminated so due to the overlapping, means one is large, another is small. As they overlap, this overlapping place 10 are reduced and it remains only 70S. Clear? All the enzymes are present, all the enzymes activity are present in cytoplasm. Plasmid contains 3 types of genes. Plasmid contain 3 genes, 3 types of genes. As we have discussed, the plasmid is present only some cell and which are known as donor cell or male cell. If the plasmid present, there are 3 types of genes may be present. First one is F gene. This F gene help in fertility, which makes the sex pili. These sex pili, large finger-like projections, are produced due to the F gene. Second one is R gene. R genes provide the resistance. This gene provide resistance or resistivity. And third one is Col gene. Col gene provide resistivity or resistance. This also provide resistance against the cholicine, a chemical substance which is found in E. coli bacteria. Col gene provide resistance to against the cholicine which is found in E. coli bacteria. So there are 3 types of genes are present in plasmid which help in different ways of the prokaryotic cell. F gene that is able to fertility, this makes the sex pili. It means only plasmid present in male cell. Second one is R gene. R gene provide resistivity against any outer chemical substance surrounding areas. And third one is Col gene. This Col gene also provide resistivity against the cholicine. Cholicine is the chemical substance or protein which is synthesized by which is produced by the E. coli bacteria. This is the structure of prokaryotic cell. As we have discussed, prokaryotic cell doesn't contain nucleus membrane, so they are called prokaryotic. All the genetic material means nuclein and cell organs are scattered all over the cytoplasm. All cell organelles are also not contain membrane, so they are not found membrane bounded organelles. Clear? One more thing is, what do you understand by inclusion body? Inclusion bodies. As we have discussed in prokaryotic, 70S ribosome present. Ribosomes are the protein synthesis factory. Ribosome consists 2 subunits. One is a large subunit and another one is small subunit. Large subunit that contains 50S and small 30S. Both the subunits are separate from each other, but in presence of magnesium ion, both the subunits are attached with each other. During attachment, some ribosomes functions sedimentation coefficient may overlap and that reduce 50 or 30S is 70S. So we can say that in prokaryotic cells 70S ribosome present which synthesize the protein. Then other one is inclusion bodies. What do you understand by inclusion bodies? There are some cell organelles except then lysosome, mitochondria, except then some granules forms which are not membrane bounded, they are also found in cytoplasm. Those are called inclusion body. For example, phosphate granules, phycocyanin granules, and glycogen granules. These are also found in cytoplasms because they are another, except then cell organelles. Some granules forms are not membrane bound organelles are also found in prokaryotic cell in cytoplasm. Those are called inclusion body, or we can say that those makes the inclusion body, such like as phosphate granules, phycocyanin granules, as well as glycogen granules. In next part we will discuss on eukaryotic cell. Hello, one more thing is remaining, that some prokaryotic cell may be stained with gram chemical substance, and if they stained, the cell wall becomes purple in color. If they are not stained, then that is called negative gram negative. So gram, what is gram positive and gram negative? Christian Gran, a scientist which show the some cells of prokaryotic may be stained or not. Christian Gran, a scientist which show that some cells may be stained. This is a stained and this one is stained, and some may not stained. The cell which stained, which colorized, those are called gram positive. That is called gram positive. And those prokaryotic cells which are not stained, which not be colorized, those are called gram negative. The cell, if the prokaryotic cell may be contain this one, and gram chemical substance is used is this one, then at the effect of gram chemical substance, the cell wall becomes purple in color. Purple in color. It means the cell is colorized and change into purple color. So this type of prokaryotic cell is called gram positive. But in some prokaryotic cell, except then, or we can say that outer side through the of the capsule, one more layer of lipoprotein is produced. One more layer of lipoprotein is produced. Due to this lipoprotein layer, the gram chemical substance is not able to stay in the cell wall, because the chemical substance not reach to the cell wall, and at that time cell wall is not stained, not colorized, and this is called gram negative. Clear?